cells are developing in the thymus, they undergo negative selection, and that is if they recognize self-MHC with self-peptides, they get deleted. And the problem is that when you have inflammation, the proteasome changes. And so people often claim that this changes the peptides that are cleaved or the specificity of the cleavage, and it doesn't. In fact, it just makes it more efficient. And the problem with that argument is that if it did change the peptides that were cleaved, then it breaks central tolerance because the T cells are negatively selected against a given set of peptides that are defined in the thymus. And this is the proteasome set of peptides, not the immunoproteasome that's generated after inflammation. So if you cleave if the cleavage pattern changes during inflammation, then you're going to have peptides that were not part of that set that were used for negative selection. And so T cells could start reacting to those. And that would then drive responses to self-proteins because they were never, the T cells that see those new epitopes were never deleted. And so that's why you can't really change the criterion for which peptides are being cleaved. Okay, so today we're going to start with MHC class 2 antigen processing and presentation. And the goals of it are very much the same as last lecture. How do you get the peptides onto, how do you generate the peptides, how do you put them onto MHC class 2, and then how are they presented to T cells? Remember that CD4 T cells are typically the ones that are going to recognize MHC2. And for the most part, they're going to do this by recognizing extracellular proteins, and they're very important for extracellular responses to bacteria. Now in this slide it says intracellular bacteria, but those intracellular bacteria had to get into the cell, so they go through the same pathways. Turns out that CD4 responses are also important for responses to viruses and other pathogens, and so we'll talk about how do cytoplasmic antigens actually get into the class 2 pathway. So when we frame this discussion, we have to keep in mind what is the point of presenting these peptides to T cells. And we covered for MHC1, you want CD8 T cells so that you can kill an infected cell. But for CD4 T cells, the goal is different. You want to present peptides to the T cells so that they can promote innate cell activation, but also direct the antibody response to be the most appropriate. And so for CD4 T cells, you don't want them to start going out and killing cells. You want them to activate other cells or control the rest of the response. So the questions that we'll cover today are sort of the same questions that we had for MHC class 1. How do you generate the peptides that then that then bind to MHC2? And then how do the peptides bind to it? And it's a little different for class 2 than class 1. And how do the class 2 with peptides get transported to the cell surface? So we first have to talk about MHC2 diversity. Remember, the MHC proteins are both polymorphic and polygenic, meaning you have multiple copies of them and they're very diverse in the population. And part of that has to do with the MHC class 1 and class 2 bind peptides differently. So we covered last time, for MHC1, the peptides are generally the right length. They're about 8 to 10 amino acids, and this allows you to sort of cap the ends or bind the ends by the MHC2. So it's a closed binding groove. MHC2 is a little different. So the peptides are typically longer, or they can be sort of undefined ends. And because of that, you can't cap the N and C, the amino and carboxy termini of the peptide. So the groove is open, allowing that peptide to sort of slide back and forth. And we'll talk about why that's important in just a second. But that fundamentally is due to the different ways in which those peptides are generated. For MHC class 1, they're cut up by the proteasome, so you have a lot of control over that, where you can define the length of how big it cuts. For MHC class 2, you're taking this from extracellular sources and it's being degraded in the endocytic pathway, and so it's you have less control of how large they are when they're being cut. And so the MHC2 accommodates that by having this open binding groove to sort of find the best fit. Okay, and so because of that, MHC2 peptides typically have undefined ends, where it's just the core epitope that's important, and there can be quite long ends sticking out from the MHC binding groove. So this peptide could continue going on quite a ways. It's just the core epitope that's the part that's being presented on the MHC2. And so when you look at the length, okay, this is the abundance of a peptide, all from the same pathogen, and it's actually from a flu pathogen, I believe, that are being presented on MHC2. And so what you do is you elute these with a weak acid that allows the peptide to come off of the MHC2, and then you use mass spectrometry to see what is the sequence of those peptides. And you can see that there's many different lengths of these, but they all share this sort of core sequence, depending on how you define it, but it's the part that's in dark or bold here. That's the epitope, and it's what's actually being presented to the T cell. And so the ends here don't really matter, okay? And so you can have ends of varying length, but the core is always the same. And this is really due to how the different way in which the peptides are generated for binding to MHC class II proteins. And so if you look at sort of how these epitopes stack up, in this case, these are epitopes from different pathogens, you can see that there is a core motif, and then there's flanking or overhang peptides that sort of stretch out from the ends of the MHC protein that can be quite different, and they don't contribute in general to recognition. Now, if you look at this closely, you'll see that because these are all from different pathogens or different microbes, that again, similar to MHC1, you have sort of a core sequence, and then you have MHC binding sequences, okay, or MHC contact anchors. And so in position 1, this particular MHC prefers a non-charged amino acid, and in the center, it prefers charged amino acids at the 4 and 6 position. And then at the 9 position, it also prefers a non-charged amino acid. So those are just the motifs, and we'll cover that in a little bit, but it's really the same, where you have parts of the peptide that are pointing down are being recognized by the MHC, and the parts that are sticking up are recognized by the T cell. Now what happens is when these peptides are generated, they sort of slide back and forth in that MHC until they find the best fit, okay? So it has an opportunity to keep trying to bind to that peptide. And that's called immune editing, and we'll talk a little bit more about that in a little bit. Okay, so when we talk about MHC class II, you have, just like for MHC class I, you have 3 MHC class I and you have 3 MHC class II alleles. But because you have two sets of those, or 2 chromosomes, then you have actually 6 copies of those. Now, MHC1 are more diverse, and those bind peptides. There are other MHC1-like molecules, sort of non-classical MHC. These are HLA, E, F, and G, and these present other types of antigens that we don't discuss in this class. They present lipidated peptides or glycosylated peptides, but we're not going to cover those because it's important just to focus on the main MHC1. Now, for a class II, they're less diverse, where they have some polymorphisms, and you have also non-classical MHC class II, and we'll talk about what those proteins do in just a second. But fundamentally, class I are more diverse than class II, but there's a reason for that. And part of that has to do with their association, right? For class 1, you have one MHC protein, so you really only have 6 total versions. For class 2, you can have more than that. So when we look at these at the genome level, right, you have the class 1 and class 2 genes are put in at the same loci as the MHC class, sorry, the class 1 genes are put in the same loci as the class 2, but they are slightly separated. So here are your classical MHC class 2 genes, DP, DQ, and DR. And here are your class 1, you have HLA-A, B, and C. Now, in reality, people do call these HLA-DP, but because it would clog up this figure by having too much text there, they just abbreviated DP. Now, the A and B here refer to the alpha chain and the beta chain. So you have 2 versions, and actually for DR, you have 2 beta chains in humans. Now, in mice, it's very similar, except we call them H2 antigens instead of HLA. And you have, you only have 2. You have IA and IE. But for both of those, you have an alpha chain and a beta chain, just like in humans. Okay, so in humans, the antigen binding or epitope binding are DP, DQ, and DR. In mice, it's IA and IE. Okay, and those are both the epitope binding. The DM here and DO, there's the rest of DO, are part of antigen loading. And so in humans, they're DO and DM. In mice, they're O and M. Okay, but for both of those, they're class 2-like proteins that help get peptides onto MHC2. Okay, so when we look at diversity in MHC class 2, we have lower levels of diversity overall, but when we look at where that diversity occurs, and this is just for the MHC2 beta chain, right, you see that all of the mutations typically occur in the antigen binding part or the epitope binding part of the MHC. So again, this allows you, because you're polygenic, you have at least 6 MHC2, and those are likely to be able to present some peptide. Okay, so you have 6 different chances in any individual to present a peptide. At the population level, though, you can see there are actually hundreds of these, and so somebody in the population should be able to make a response and survive an infection. Now, just like at the genetic level or the chromosome level, the A and B here refer to the alpha and beta chain. So for DP, right, you have alpha and beta. For DQ, you have alpha and beta, and then DR, you have alpha and beta. Okay, so just like MHC class 1, variations in MHC class 2 mostly occur in the peptide binding sites. Now, that's one selection for people who can actually present peptides, but you can imagine that if you started making mutations in the alpha 2 or beta 2 regions, which are just structural, that structure would fall apart, and so you would select against people who have mutations there. But here's one extra twist, right? Because MHC2 is less polymorphic, there's less variation in the population, you actually have a trick that they do where they'll start to mix and match. So for MHC class 1, if you have 2 different variants of MHC class 1 at the B allele, HLA-B, you can have 2 different MHC1. You have 2 different variants at the C allele, you'll have 2 different MHC2, and same with A. But for class 2, you can have variation where, because you have 2 copies of DR alpha and DR beta, you can have DR with alpha and alpha from allele 1, and you can have DR with alpha and beta from allele 2, in this case A and B. But those can also, the DR alpha from one allele can pair with the DR beta from the other allele. And so because of that, for MHC2, you actually have at least 12 different MHC2 variants, and possibly more if you could have, for example, a DQ alpha binding with a DR beta, okay? And that has been shown to occur, but primarily the main variation occurs where you get reassortment of the DR in any individual cell. And so what does this tell you? Well, you end up with many more possible binding motifs. Instead of just 6 chances to present an antigen, you now have 12. Okay, so even though there's less genetic variation, there's more possibilities or more combination variation.
So does that mean that MHC2 is more polygenic versus polymorphic?
Well, in terms of polygenic, you still have three alleles on one chromosome and 3 on the other. But in terms of functional expression, you get more versions of that on the surface. It's not really more polygenic, but it's sort of analogous to that. Polygenic is not the right word because that refers to the genes. But on the cell surface, yes, you do get more variation. You get at least 12 different versions. All right, any other questions on that before we keep going? Okay, so the first question we had is how do you get these peptides? How do you generate peptides for binding to MHC class II? Remember, these are coming from extracellular sources. So the cell is taking up proteins or pathogen or some version of an antigen. And that endocytic vesicle is then fusing with a phagolysosome, which contains some special enzymes, and then degrades those antigens into peptides. Those peptides are then loaded onto MHC class II, and by doing that, allows those MHC class II to get to the cell surface. So the MHC2 is, just like other proteins, is made in the endoplasmic reticulum. It's trafficked through the Golgi, and then it fuses with the phagolysosomes. And so you have sort of 2 pathways that come together to allow those peptides that are being degraded to be put on MHC2. So the first steps are the microbes, or antigens in this case, are broken down into phagolysosomes, and then they're loaded onto MHC2 proteins, and we'll actually go on how they do this because it's not just simply floating around and binding. And then once they bind, it's transported to the cell surface. So how does this work? And we'll start with just taking in some antigen or pathogen that's taken from the extracellular space into an endocytic vesicle. Okay, now one of the first steps is that this endosome fuses with phagolysosomes, okay? I should say fusion of endosomes with phagolysosomes. And those phagolysosomes contain proteases. Now, as it's coming in, early on, that is neutral pH. And so, sorry, I'm messing this up a little bit here. Okay, so those lysosomes fuse with the phagolysosomes, and the phagolysosomes contain low pH, or they have low pH. That low pH activates the proteases that are in the endosomes, and that allows them to start cutting up the proteins into smaller peptides. Now, the next step is once these cut up peptides in the endocytic pathway are going to fuse with the specialized exosomes containing MHC2. And this is called the MHC2 loading compartment, or the M2C. That was discovered by Jerry Boss at Emory in the late 90s. And what happens then is that there is loading of the peptide onto MHC2. It's actually an exchange of peptides though, because remember, a peptide is integral to the MHC structure. There's no peptide there, the MHC falls apart. So have to have a peptide. So in this case, it's exchanging one peptide that the MHC2 is synthesized with for another peptide that is brought in. So let's talk about the proteases, right? Here are the proteases that are being activated by low pH. And then typically we call those the cathepsins, okay? Now cathepsins are not unique to the immune system. They're actually expressed in many, many types of cells. And they're really important for other things like recycling proteins, for maintenance of neurons. So they're ubiquitous, okay? And the first one is cathepsin B, whose job is to use a cysteine thiol. I'm actually not showing the thiol part here, but there's the cysteine part of this that does a nucleophilic attack on the amino acid bond, okay? Do I care that you have to know the electron transport or the electron attack here? Not really. What I'm more interested in you knowing is that it's cleaving at the peptide, at the peptide bond, so you're actually leaving the N-terminus of the peptide. So the cathepsin B is interesting because it typically is measuring away or a distance from the carboxy terminus of the protein. And it'll typically cleave between 9 and 18 amino acids from the carboxy terminus. And so you can get rather long peptides that are being cleaved here. Cathepsin B is present in all of your endocytic compartments. It's not just the phagolysosomes, but it's activated in the phagolysosomes. So this allows you to sort of control where are you going to have this degrading your cell proteins. Now, cathepsin B is also present in the exocytic compartment. So it's responsible for cleaving prohormones or proenzymes into their active form before they're released from cells. So it has other roles besides this antigen generating part of the immune system. And for any of you who have ever tenderized meat in pineapple or citrus fruits, you're actually using cathepsins. It's high, particularly in citric fruit or citric juices. So if you incubate your pork loin in pineapple juice, it's actually degrading those proteins to some degree, allowing it to be tenderized. Just trivia there. Okay, so the first one is cathepsin B. The second one is cathepsin D, and I put on here and E. There are other cathepsins, but the main one is cathepsin D. And it uses a different cleavage pattern. It's actually using an aspartic acid carboxylic group with a water molecule, and that's actually the electrons from the water molecule are again attacking the peptide bond. Okay, so you're generating amino and carboxy terminus. Now, cathepsin D doesn't cleave the same way as cathepsin B. It actually has a preference for cleaving after some hydrophobic residues. So it has more sort of random cleavage patterns. It's not measuring from the end, oh, I want some distance of peptide. So because of that, cathepsin D is thought to be less important than cathepsin B, but still does play a role. And it also has other roles outside of the immune system for maturation of prohormones or proenzymes that are being secreted. And so if you don't have cathepsin D, you can actually get fatal neurological disease where you're not releasing the right neurotransmitters, and so neurons fail to develop properly. But in mice that don't have cathepsin D, it doesn't look like you have much change in antigen presentation, suggesting it may not be that important, or that other cathepsins like cathepsin E may also do the same thing and sort of compensate for it. But in general, cathepsin B is typically more important, and then cathepsin D does play a role. Now, if you think about proteins, right, you have sort of their linear sequence with their peptide bonds. But remember, they also get folded into shapes by, in particular, having disulfide bonds, right, between cysteine residues or the other sulfides. And so in a normal protein, you have this disulfide bond. And so you don't want to present, if you can imagine if you presented a peptide that was stuck to another peptide by a disulfide bond, you'd actually have this kind of weird complex on the surface. So to take care of that, you have to reduce that disulfide bond. Okay, now there's a couple of things that happen. First, the phagolysosomes have low pH. So that favors a reduction of the cysteine bonds, but it's not enzymatically very favorable. And so in the MHC2 compartment, you're also expressing a protein called GILT. This is interferon gamma, so the G is from gamma, inducible lysosomal thiol reductase. And so there's, this was originally discovered in Pete Creswell's lab, but is now being followed up by people at U of A downtown Phoenix. And essentially, it enzymatically reduces this bond to free the sulfurs. So the low pH is interesting, right? Because it activates the proteases, but it also is inactivating them, where it eventually chews those up. So you have sort of this limited window of digestion, but then you have to reduce the disulfide bonds, which are internal to many proteins. And so cysteine reduction is probably the last step, where it's actually expressed in the MHC2 compartment after the peptides have been already degraded. Okay, so any questions on how we generate the peptides for MHC2?
I'm sorry, I'm a little confused. Are phagosome and endosome interchangeable?
Well, the endosome is actually the compartment that's being taken in from the extracellular space. The phagolysosome is actually a compartment that's it's actually exocytic, right? It's being put out towards the surface. And so the endosome is endocytic, the phagolysosome is exocytic because it's fusing with that as it's still being brought in. So it's sort of like getting intercepted.
Thank you.
So you sort of have endosomes that fuse with phagolysosomes that then fuse with the MHC2 specialized compartment. So there's actually two different fusion events happening here. Any other questions? Okay. So now we've generated peptides, right? They're in the M2C compartment where the MHC is. How do they bind? And remember I said that when you make an MHC protein, or when these are synthesized in the ER, they have to have a peptide bound to them. And so remember for MHC class 1, you had these chaperone proteins that were sort of holding it there in the absence of a peptide. For MHC2, you have a specialized chaperone protein called the invariant chain. And this is often just put as big I, small I. And there are historic reasons for that, but that just means invariant chain. Okay, and this was identified also by Pete Creswell at Yale, who spent a semester here last year. Maybe it was a little longer than that. So what happens is these chaperone proteins help direct folding of the MHC2, but they also have an arm that binds into the MHC2 peptide binding group. Okay, so the cytoplasmic tails target this MHC2 to the specialized compartment called the M2C, and then they also direct the folding of it, and they provide that initial peptide to make sure that the MHC2 is folded, that it doesn't fall apart. Okay, and if you don't have this MHC2 protein called the invariant chain, then you don't get any MHC2 on the cell surface. Now what happens is that the invariant chain, II, is degraded, right? As it is being put out towards the cell surface, and it fuses, this M2C fuses with the phagolysosome that is fused with the endosome. Stay together with me. I know there's lots of different compartments here. You initially have the invariant chain is bound to the MHC and it's directing where it should go. But then as it's binding, or sorry, as these two compartments are fusing together, that invariant chain is degraded. And what's left is just the peptide part called CLIP that remains bound to the MHC2. Okay, so once the cathepsin S and L that we haven't talked about that are in the M2C, these degrade the invariant chain specifically. That's their main job. Okay, and their job is to cleave this invariant chain, leaving just the CLIP protein still bound to MHC2. Now, the terminology gets a little confusing here, right? II is the H2IA, right? That's one of the mouse MHC associated invariant chain. CLIP was originally called the class 2 invariant chain peptide, but that actually, many people will say it's Cresswell's little invariant chain peptide. And so immunologists are terrible at naming things. I don't care which version you know. I talked to Pete last year and he said he never called it Cresswell's invariant chain peptide. So the official version is the class 2 invariant chain peptide.
So, Dr. Blattman,
Yes.
is the invariant chain then kind of synonymous to the TAC for MHC1?
It would be more, it's not terribly synonymous with any of them because it is not really transporting peptides, which is what TAP does. It is a chaperone protein, so it directs folding of the MHC2. So you could definitely call this something like calreticulin, right, where it's directing folding. But then it has this other structure part of it that's bound to the MHC2, and there's not a comparable version in MHC1 compartment. Okay, so now you have CLIP, or you have the little CLIP peptide that's bound to MHC2, and it fuses with this endocytic compartment that has a degraded peptides in it. And how do you get those peptides onto it? And so we talked about these other non-classical MHC class 2 proteins. And the first is DM, HLA-DM. And the job of HLA-DM is sort of analogous to ERP57 and calreticulin, where they're sort of holding it open. But DM is basically coming in, knocking MHC2 open so it can release CLIP, and then other stuff can bind. Now, DM is a protein that's not expressed at the cell surface. It's only in this M2C compartment. And its basic job is just to hold the MHC2 open for long enough that it can exchange the peptide it has bound for another peptide. And as we'll see later, it does this many times, and so you get the best fit peptide. And this is how you allow that peptide to sort of slide back and forth and find its best binding group, because DM keeps opening the other HLA class 2 molecules. Now, what happens then is you edit the epitopes that are being presented by MHC class 2. Okay, so it's not just once that DM is coming in and opening that MHC2. It keeps doing this repeatedly. And so each time, even if that MHC2 has a foreign peptide on it, it can still edit that and bind a different one that if something else binds better, then you exchange one peptide for another. And this process is called epitope editing. And it's the way in which you select the highest affinity peptides for binding to to MHC2 proteins. And so once they get on the surface, you've sort of sampled that, representing your best epitopes. Now, there's another protein that was discovered by Brian Evevold and others called HLA-DO. And by the way, if I mention somebody's name and I didn't put their picture or put their name in the slide, you don't have to remember the name. Okay, it's easier for me to keep it straight by remembering who did what. But in any case, what Brian showed is that you actually needed another protein, HLA-DO, for this for HLA-DM to work. Okay, so DM's job is to open up the MHC2 and release the CLIP peptide. getting a little crazy with our animations here, so that other peptides can bind. But in order for it to do this, it has to cause a conformational change. And if you think about protein structures, for most proteins, they get one shot at a conformational change and they can't change back. But DM has got to do this repeatedly over and over. And so DO is not the thing that's facilitating peptide exchange. Rather, it's a placeholder for the MHC2, and it resets it. So each time that DM interacts with an MHC2 and causes that peptide to come out, so here the peptide's coming out, then it's got to reset. In order to do that, it has to get back together with DO. Right? And I won't commentate on how this might be like some of our social lives, but you sort of have to reset. Okay? And the job of DO is to reset DM so that it can come in and do this again. Okay, so any questions on how the peptides get loaded onto MHC2 proteins?
I do have a couple of questions actually. So does every time that DM loads MHC2, does it have to bounce back to DO?
Yeah.
It's just constantly bouncing back and forth.
Yeah, it's going back and forth. It has to interact with the MHC2, go back to DO, get reset, then it can go back to the same MHC2 or a different one, and go back to DO and get reset.
And then, so CLIP after S and caspin S and L degrade that, it's long chain. Is it realistically just a placeholder at that point, or does it have any other role?
As far as we know, it's only a placeholder. And there are mice that that have a modified MHC2 that the clip remains bound. You can't release it because it's tethered to the MHC2. And those mice develop fine. So there doesn't appear to have any other role except just as a placeholder.
Dr. Blattman, are those mice that you're speaking about, are they like knockouts of DM or DO? Is that why they still have the clip bound to the MHC2?
No, it's a modified MHC2 in which the clip protein is actually part of the protein sequence. And so it is covalently linked to the MHC2. We might still have DM and DO, but because it's covalently linked to the MHC2, it doesn't get released. And then people have used those mice to try and figure out things about negative selection. So what happens if you select for T cells and there's only one peptide on MHC2? Do you get autoreactivity or other things? And the answer is yeah, it is a big problem.
Dr. Blattman, how does it know that it's selecting for the best peptide?
You don't necessarily, it doesn't know. If we can't really anthropomorphize it, right, where we're saying it knows. But what you're doing is you just keep opening it and allowing the best one to fit. And so sometimes it won't be the best one. You know, it's got a limited time, but you're just doing it so you have the best chance of binding the best peptide. So you can imagine first, if it opens up and has something there that binds, but it has a really low affinity for MHC binding, when it opens up again, the chances of that coming out are pretty high. And so something else can come in with a higher affinity and bind. And then if that gets opened up by DM again, it's less likely to come off. But it could, and something else with even a higher affinity could come in. So it's sort of just selecting for things that are binding strongly. Any other questions? All right, let's keep going then. Now, the problem is that you need CD4 T cell responses to viruses. You need CD4 T cells for intracellular bacteria. Often CD4 T cell responses are directed against internal proteins that are never actually seen outside of the cell. So how do you do this? How do you recognize it? And remember, when we talked about MHC class 1, you have to make CD8 responses against some extracellular things or some things, proteins that are being outside the cell. So we had this cross presentation, okay? And so the reverse of that is how do you get peptides that are cytoplasmic, as in viruses, how do you get those into this M2C pathway? And the answer is a process called autophagy. And autophagy has many roles in cell biology. It's not strictly the immune system, but we're going to talk about how autophagy allows you to actually make these responses to, how you make CD4 T cell responses to internal proteins. Now, in cell biology, you probably have heard about autophagy and sort of the recycling of organelles or proteins. And in particular, macroautophagy is this process where you take things like a mitochondria in cases where you have low nutrient supply. The mTOR pathway is activated, and then you start this autophagy pathway. And there's a number of cells that do this macroautophagy. But essentially what happens is you're taking a bilipid membrane or an endosome and sort of folding it over on itself. And it's sort of like if you had a garbage bag that you trying to wrap a garbage bag around something, it's got 2 layers around it on all sides, right? And so when you do that, these autophagosomes are directed to lysosomes or phagolysosomes, and then those organelles are broken down and recycled. And this uses a lot of the same things, right? You're using cathepsins, you're using GILT, sort of the same proteins that you're using in the M2C compartment, but they're degrading these things. And so you can potentially load peptides derived from intracellular sources into the M2C compartment and load them on MHC2 proteins. Sorry, if the terminology gets too much, just let me know, okay? I'll try to make it as clear as possible. But this is really not that efficient when you're talking about viruses or small particles. It's really sort of more designed for large organelles that are being engulfed by these autophagosomes. And so what you have that's perhaps more important for the immune component of autophagy is you have the lysosomal associated membrane proteins, the LAMP proteins. And these are membrane-bound proteins that are associated with the lysosomes, or they're in the membranes of the lysosomes that are that are fusing with the endosomes of whatever you're bringing in, okay? And so one of their jobs is to transport antigens from the cytosol. So in this picture, the cytosol is up top and the inside of the lysosomes on the bottom. They're taking proteins and they're directing their transport into the inside of that lysosome. Okay, so this has got a lot of detail on it. I don't care that you know all the detail. It's just basically you have a chaperone protein, which these are called the heat shock proteins. And this will bind to misfolded proteins or proteins that have been labeled with ubiquitin, take them to the LAMP proteins, and put them into the lysosome intracellular, or the inside of the lysosome. So you can think about this as sort of a reverse TAP protein. Well, actually, it's a TAP-like protein. Doesn't look to have the same structure as TAP, but it's functionally, its job is to take stuff from the cytoplasm and to direct it into the lysosomal compartment, okay? And then once this happens, the LAMP proteins are actually degraded and they're transported along with everything else to the cell surface, or they get recycled. I don't care that you know all of the steps in this pathway, just that there's a protein, HSC70, that's recognizing some cytoplasmic proteins. It takes them to LAMP, and then LAMP transports them into the lysosome. And eventually they get degraded, but I don't care so much that you know about the degradation of those, because that's not the important part. This is more about how do you get epitopes from viruses into the M2C. Okay, so here we have the MHC class 2. It's bound to the invariant chain, which is directing its migration out. We've already talked about extracellular things are brought in, the early endosomes. These fuse with the phagolysosomes, and then also with the outcoming MHC2 to form the M2C, okay? And it's here where you have degradation of the invariant chain, leaving clip, and you have the chance to load the degraded peptides that are coming in. But from autophagy, right, either through macroautophagy or perhaps more importantly through LAMP protein transport, you also have peptides put into this compartment that is coming to the M2C. And so you can have things from the cytoplasm that are brought out in the autophagosome, or typically we'll call this the phagolysosome, that then when they fuse with the M2C, now you have sources of intracellular peptides, whereas from this pathway, you have extracellular peptides. So that allows you to make CD4 T cell responses to intracellular pathogens like viruses. If you didn't have this, you wouldn't be able to make antibody responses to viruses because you wouldn't have CD4 T cells. So it's essentially the analogous process for cross-presentation, right? Cross-presentation is when you take extracellular proteins, you transport them into the cytoplasm by Sec61, and then those get degraded by the proteasome. Where in autophagy, you're taking cytoplasmic proteins and you're transporting those into the autophagosome by the LAMP proteins, and then those are fusing with a compartment where you're loading class 2. So you get cytoplasmic proteins onto class 2. Okay, any questions on autophagy? Do I expect you to know all the proteins and macroautophagy? No, there's a ton. That is the realm of cell biology, and I think would just slow us down in learning immunology. Do I expect you to know the proteins for microautophagy, which is essentially LAMP transport? Just the HSC70 and the LAMP2 protein. I don't expect you to know all the other associated proteins, of which there are many. Okay, if there's no questions, let's keep going. Okay, so now we've got peptides that we generated either from extracellular sources or intracellular sources. We put those onto MHC2 in the M2C compartment. We essentially exchange the clip for one of these other peptides. And that's done by DM, bumping it open, getting reset by DO, going back. And so you get the best peptides possible bound, or the highest affinity peptides bound. And then that cell traffics to the cell surface. The question is, what do you want it to do? Okay, so there's a lot like, much like CD8 T cells, your initial activation of CD4 T cells has to be on professional antigen presenting cells. Now, in many books, including this textbook, they'll say, well, what are those professional antigen presenting cells? Initially, it is all dendritic cells, okay? Only dendritic cells can start a response. But as the response keeps going on and you get production of cytokines by the CD4 T cells and others, you can have macrophages and B cells, as well as other T cells, can all become professional antigen presenting cells, okay? So initially, it's dendritic cells. So you would put here, the very first ones have to be dendritic cells, but then it can be macrophages and B cells. And these are going to further activate the T cells. And we'll talk, in the next section of the course, we'll talk about all the different things that T cells will do including this continued help for B cells, then the job is really to activate these cells. Okay, so dendritic cells activate the T cells, and then the T cells are activating macrophages and B cells. Okay, so that's why here it says it's presented to effector T cells. Where did those come from? It came from up here, from the dendritic cells. So what does it do? Well, if a CD4 cell is going to recognize a macrophage, it wants to enhance the activity of that macrophage. It wants to make it better. And so what it'll do is it will activate the macrophage to kill anything that's inside of it, okay? And its job is, and that does it primarily through interferon gamma and IL-12. And so this is important for things like TB and other bacteria where the bacteria can kind of hide out in the endosomes of the macrophage and just not be recognized. Well, if the T cell recognizes some antigens on that macrophage, then it will make cytokines that will tell that cell, yeah, you're infected. You need to start killing what's ever in your endosomes. Now for B cells, it's different. The CD4 T cells are going to come in and recognize there's antigen on a B cell. But if you think about it, B cells are bringing in antigen by their B cell receptor, meaning they're antigen specific. And so the T cell is not going to say, oh, you're infected. Instead, what it's saying to that cell is, you brought in antigen, you presented it to me, here are your activation orders, go and make antibodies against that, okay? So the goal is a little different for the different cell types, but the main job of CD4 cells for B cells is to tell them, yes, make an antibody response, and then also it'll tell them what kind of an antibody response. Should it make IgG? Should it make IgE? What is going to be most effective? Okay, so you can imagine just for like MHC1, right, where viruses have figured out, well, if I just block MHC1, I can hide out from the T cell response. It should be no surprise that viruses have figured this out for MHC class 2. So for HIV, it makes a protein called NEF, and part of the job of NEF is actually to bind to the MHC2 and retain it in the lysosome so the MHC2 is degraded and never gets to the cell surface. For other viruses like cytomegalovirus, right, it just makes a protein that prevents the MHC2 from getting out of the endoplasmic reticulum. So it never leaves. Other proteins like the A35 protein from vaccinia virus will actually block fusion of this incoming thing, or your endosome here, or block fusion of the autophagosome that develops from actually going to the Golgi. Sorry, it's not entirely correct. It's not really Golgi, it's fusion with the M2C. It's not full, this must be from an older figure. It's my mistake. Okay, so most MHC2 inhibition, because remember for MHC1, what does it block? It can block transport of the MHC1, but a lot of the times it'll block TAP, or it'll block sort of the initial steps of generating antigen. It'll block the proteasome, it can block TAP. It's much harder to do with class 2 because you're talking about just taking stuff in and digesting with proteases. So instead what it does is MHC2 inhibition is blocking the transport. Now the other thing that that viruses will do is will suppress the signals that tell a macrophage or a B cell to express high levels of MHC2. Okay, so there are some examples of blocking transport of MHC2 inside the cell, but most of the time viruses are trying to interfere with signals that would activate a cell to become an antigen presenting cell. So they block macrophage and B cell activation, so you don't really ever see enough MHC2 on the surface. Okay, so what do I expect you to know? Okay, so we go back to our questions about what do we want to know about MHC2? I want to know, how do you generate the peptides? So you should be able to tell me, what do cathepsin B and D do? What does GILT do, right? What are their roles in degrading the proteins in those endolysosomes? And for autophagy, what is macroautophagy, but also what is this LAMP transported dissociated version, which you take cytosolic proteins and feed them directly into the lysosomes. Okay, so you should know those two pathways. For macroautophagy, you don't have to know all of the autophagosome proteins that are generated to sort of generate the surface. But then you need to know how do the peptides bind to the MHC2 and how do they get transported to the cell surface? So you should know something about this difference between MHC1 and 2, where MHC2 has core epitopes that can have long overhangs that stretch out of the MHC itself. So the anchor residues are typically just in that core epitope. And then you should be able to tell me, what does the invariant chain do? And that it's degraded by cathepsin L and S, although I just said I wouldn't ask you on that, so let's take that one out. And then what is the role of CLIP, HLA-DM, and HLA-DO? Right? CLIP is the part of the invariant chain that remains bound to the MHC2. DM allows you to exchange that for other peptides, and DO is resetting DM. So let's have some in-class questions. Okay, and these are not in your slides because I want you to think about them. Okay, and just please, you can answer verbally or just, you can answer in your own head. The questions are these. If I make a CD8 T cell response to Salmonella typhimurium, is the CD8 T cell response going to recognize MHC1 or MHC2? Okay, so people are answering by chat, that's fine. And so far everybody's right, it's MHC1. CD8 T cell response, CD8 T cells recognize MHC1. Now the second question is, where do the antigens come from? Is this classical antigen processing and presentation or cross-presentation? So far, only one person is brave enough to answer. Where does Salmonella live? Does it live in the cytoplasm or does it live outside the cell? So it mostly lives outside the cell. So in order for it to be the classical pathway, it'd have to be in the cytoplasm. So you need that extra set of taking it extracellular proteins, feeding them into the cytoplasm where they get degraded by the proteasome, and then put back out by tap into that. So that's cross-presentation. So whenever you have an extracellular thing, it's cross-presentation. If it's cytoplasmic, it's just a classical pathway. The only difference is that additional step by Sec61 by putting it into the cytoplasm first. So, do you require the proteasome for this? Okay, see, people are starting to say, yeah, it's still proteasome dependent. It's always proteasome dependent. You can't, if you feed that protein into the cytoplasm, it's not going to work unless it goes through the proteasome that then gets pushed through tap, okay? All right, same infection, but now we're talking about the CD4 T cell response to Salmonella. Is it MHC1 or MHC2? Right, it's MHC2. CD4 T cells recognize peptides on MHC2, so that doesn't change. Is this the classical pathway or autophagy? Okay, so people are chiming in, everybody's right, it's classical pathway. The classical pathway is taking extracellular stuff. So remember, salmonella is outside the cell. And the classical pathway is taking stuff from outside the cell into that endosomal compartment, degrading it when it fuses with the phagolysosomes, and then putting it in the M2C compartment where you can put those degraded peptides onto M2C. Autophagy would be for things that are inside the cell. So you don't need autophagy here. It's just the regular classical pathway. Now, is this cathepsin dependent? Okay, so I see people responding, yes. Well, absolutely. In order to degrade these, you need cathepsins. And in fact, even for the other pathways of autophagy, you still need the cathepsins so that you can degrade the proteins. If you don't have those, it'll just be a big chunk of protein and you can't present it on the MHC2. Okay, we're all doing very well so far. Okay, so this one gets a little trickier. I'm going to ask you about the CD8 T cell response to your first dose of measles vaccine. Is that going to be classical pathway or cross-presentation? Okay, so we're all saying cross-presentation. Well, that's probably true for the initial step, the very first activation. And as we're getting to a gray area, the very first cells to be activated are probably by cross-presentation. It depends on a lot what the measles vaccine is, okay? In this case, the measles vaccine is a replicating thing. It's an attenuated virus. So it does get in the cytoplasm and it does replicate. So in reality, a lot of it, the vast majority of the response is the classical pathway. Having said that, you need that first step to be cross-presentation and then the classical pathway kicks in. So it's a bit of a trick question there, but luckily I don't do that on exams. But what about the CD8 response to the booster vaccine? Right, it's the classical pathway. These cells have been licensed. They already know the things are there. We'll go into immune memory and sort of what happens, but it's basically don't need the cross-presentation to start everything. You've already got the cells you need. Okay, now I know that those last questions were a little bit more vague because of, I'm sort of getting you to think about when would you need classical presentation versus cross-presentation. In the first dose, probably to start it, you need cross-presentation. If I frame that question a little better, I can make it clear that's just cross-presentation. But in a booster situation, it's all just classical pathway, so you don't need cross-presentation. All right, so that's all I have for you today. We're going to end a little bit early because I know this is a lot of information for you to be studying for on the test. So I want to have open up for time for you to ask questions now.
So Dr. Blattman, for the CD8 T cell response to salmonella, that one's the cross-presentation one because it has to be brought inside from extracellular.
Correct. Right. It's extracellular, so you have to put it in the cytoplasm somehow.
Dr. Levin, so with the M2C, is that moving around and binding to a bunch of different phagolysosomes, or the phagolysosomes heading to the M2C, or are they kind of just on a cross collision? And if there's nothing there for MHC2 to like recognize or to present, does it then like disconnect from the fatal lysosome and go to a different one?
So the first part of your question is who's headed towards who?
Yeah.
And that's not entirely clear because it's difficult to figure that out, right? But you can think of them as going to the same place, so they're on a crash course for each other. I think that's the best way to remember it. I don't care that you know who's moving towards who, just that they fuse together. Okay. And the second part of your question is, if there's no peptides, what happens? Is that right? So
yeah.
Well, remember, it's still got clip. Okay, so there's at least that's there. But in reality, this process is going on all the time. And so even if there's not an infection, you're still putting self-proteins into that compartment, right? You're still sampling the environment and you're still degrading whatever you take in. So there's always going to be some peptides there. But even if there wasn't, you'd still have clips, so it'd still go to the cell surface.
Okay. So no matter what, the MHC2 wouldn't just get degraded there, it still would head towards the cell surface. Correct.
Now, another part of your question was, does it diverge from the M2C and then go and find another phagolysosome? The answer is no. You can't go backwards in that fusion step, because how would you sort out which goes to where?
Okay, that makes sense. Thank you.
Dr. Blattman, I've got a question. Is there anything particularly interesting about DM or DO? Like, is there any pathology known about or related to like lack of DM or DO and anything about that?
Well, I'm not sure in humans, but in the mice that these were discovered in, they discovered that that they basically just all had clip on the cell surface. So it was a mutant strain of mice. I don't know that there is a human equivalent that there's a disease pathology known for. I could look that up and answer that next class, but I don't know.
Okay, and also, are there any particular cells that express more DM or DO, or is it pretty like evenly dispersed, kind of?
Well, remember, the cells that start the response are dendritic cells. So when a dendritic cell gets activated and it travels to a lymph node to start a response, it has high levels of DM and DO. Once you start that response and you get cytokines that are activating B cells and macrophages, they'll also express DM, DO. But class 2 is limited just to hematopoietic cells. Okay, you don't express class 2 on just willy-nilly anywhere in the body. It's very limited to just hematopoietic cells that can act as antigen-presenting cells. And because of that, DM and DO are really only expressed in those same cells because you don't need it anywhere else.
Okay, thank you.
Professor, you have a question in the chat.
Does cross-presentation pertain to intracellular proteins? So cross-presentation is taking extracellular proteins and transporting them into the cytoplasm and then degrading them. So cross-presentation is just the classical pathway of that one extra step of taking extracellular things into the cytoplasm. So once it does that, they become intracellular proteins, but yeah, it's for extracellular things. Other questions? So the question then is, did I confuse you on this question? And it really comes down to the first dose of the measles vaccine. To get things started, you really need cross-presentation, okay? Because you have the dendritic cell that's sitting out in your vaccination spot. So in this case, your derma is going to take up stuff from the extracellular place. It's doing this penocytosis and recognize there's something here, travel to a lymph node, and then present whatever it took up from that extracellular space. So even though this is a virus, initially to start the response, it's mostly cross-presentation. That's what activates T cells. Now many would argue, well, some of the dendritic cells get infected, and so it's probably also cross-presentation and some classical pathway. But in general, most of the times when you start a CD8 response, it's mostly from cross-presentation because it's a result from that dendritic cell doing penocytosis, which is by definition extracellular or extracellular junk.
Dr. Blattman, I was confused about this question because I was thinking, because it's a vaccine and you're going to be getting this administered via like a shot, I was thinking that maybe it would be more like intracellular or something.
Well, for it to be intracellular, it'd have to be infect a cell and then start replicating and express proteins. So the first step of a response is remember dendritic cells are sitting in the periphery sampling their environment, right? And they're doing that by taking things in endosomes through this penocytic pathway. And they're going to then, remember there's endosomes can recognize things that get degraded there, including viral nucleic acids. That signals a dendritic cell, yep, you've taken something in, go to the lymph node and start a response. So that first step is usually cross-presentation. Now you're right in saying, well, okay, this is a replicating virus, so it could infect a dendritic cell, but so that could happen as well. But it's usually, most of the time, it is this cross-presentation because they've taken it up in penocytic vesicles. So the next question is during autophagy, do you get self-proteins binding to MHC2 and eventually presented on the surface of the cell? The absolute answer is yes, absolutely you do. If there's nothing infecting that cell or it hasn't taken up anything, the only thing that could be presented are self-peptides. And those are going to exchange for CLIP just the same way as anything else. Other questions? Okay, well, study hard. We'll see you on Tuesday. We will be available for either email or I think we'll try and have the Zoom room open in case people need to log in and ask specific questions.
No bonus question today, right?
No bonus question today. All right. Thank you, everybody. We'll see you on Tuesday.
Thank you.